Hello everyone, I am Srivalli, a member of Shivi Forensics. Today, in this video, we will see 10 MCQs from Forensic Serology and the reasons behind their answers. The first question is, what is the primary function of hemoglobin in blood? Option A, digestion. Option B, oxygen transport. Option C, blood clotting. And option D, immune response. The answer is option B, oxygen transport, because hemoglobin binds to oxygen that is present in our lungs to form oxyhemoglobin and carries it throughout bloodstreams to cells which requires aerobic respiration. The next question is, which of the following presumptive tests is the most reliable technique to analyze a given substance as blood at a crime scene? Option A, luminol. Option B, LMG test. Option C, Castle-Meyer test. And option D, acid phosphatase test. The answer is Castle-Meyer test, which is also known as phenolphthalein test, because when compared to other presumptive tests, the Castle-Meyer test is the most reliable, mainly due to the easy availability of the reagents and the easy transportation of the same reagents, so that we can test whether a substance is blood at a crime scene itself instead of transporting the substance to forensic labs and having to test it. The third question is, what are the main functions of genetic markers in forensic investigations? Option A, determines ancestry. Option B, determines blood type. Option C, individual identification. And option D, measure blood glucose levels. The answer is option C, individual identification. Based on the presence of genetic markers in biological samples like saliva, semen, blood, urine, etc., we will identify individuals by DNA analysis, because genetic markers differs from person to person and are not the same. The fourth question is, bloodstain pattern analysis can help to determine option A, height of the suspect. Option B, direction of blood movement. Option C, genetic profile of the suspect. And option D, exact time of death. The answer is option B, direction of blood movement. The analysis of bloodstain pattern would help us to determine the direction and the angle of impact, the type of weapon used, and the distance between the victim and the assailant, which will serve as a vital evidence in forensic science and help to reconstruct crime scene. Other than this, height of the suspect, genetic profile of suspect, and the exact time of death cannot be determined with bloodstain evidence. The fifth question is, which test is commonly used for the identification of saliva in forensic cases? Option A, acid phosphatase test. Option B, Takayama test. Option C, heme agglutination test. And option D, Fedebas test. The answer is Fedebas test. This is a presumptive test for saliva, which basically uses dye-labeled starch tablets that react with salivary amylase and releases color upon by the process hydrolysis. The next question is, how can Takayama test be classified within the hierarchy of forensic blood analysis? Option A, confirmatory crystal test. Option B, individualization method. Option C, species origin test. And option D, preliminary presumptive test. The answer is option A, confirmatory crystal test, because confirmatory test of blood like Takayama and Takman are also known as heme crystal assay, as the reagents that we use for the test reacts with the hemoglobin content in our blood to form highly distinct and unique shaped crystals. The seventh question is, what is the confirmatory test in which Christmas tree stain is used? Option A, acid phosphatase. Option B, creatinine. Option C, casein. And option D, alpha amylase. The answer is option A, acid phosphatase. This is a genetic marker which is present in seminal fluid. So, when we use the Christmas tree staining to stain spermatozoa's head and tail with different colors, acid phosphatase reacts with the Christmas tree reagent and will help us to detect the spermatozoa in the semen sample. The eighth question is, which of these factors affect the drying time of a bloodstain? Option A, surface texture. Option B, blood volume. Option C, environmental conditions. And option D, all of the above. The answer is all of the above, because based on the volume or the quantity of a stain, and what surface it is present, whether a surface is porous or non porous, and the environmental conditions like humidity, temperature, and how the climate or the weather is like will all affect a stain on how much time it takes up to dry. Ninth question is, which type of bloodstain pattern results from the force of gravity alone? Option A, impact spatter. Option B, passive drops. Option C, cast of stains. And option D, expirated blood. The answer is option B, passive drops. Expirated blood is something that comes out from our bodies like the nostrils or the mouth, while impact spatter and cast of stains require mechanical forces like the type of weapon a man is using and how many hits the assailant is giving to the victim and how the weapon is swung by. While passive patterns like drop, drip and pool of blood or flow of blood does not require any mechanical forces, and they are created solely using gravitational force and not by any other external force. The last question is, what is the widely used method that helps to determine the species of blood? Option A, precipitin test. Option B, electrophoresis. Option C, blood grouping. And option D, enzyme typing. The answer is option A, precipitin test. This is based on the antigen antibody reaction, which is basically present in the blood, and that antigen antibody reaction will help to determine whether a blood sample belongs to a human or an animal, while the other test like blood grouping will simply tell what type or what kind of a blood, like if that blood group is from the group O or A or type AB, like that. Thank you for watching. Make sure to subscribe Shivi Forensics and click that bell icon so that you receive all the notifications and our newest videos.